Okay, the thing is, in poorer countries, generally, meat is made from ex-work animals, cattle which have been used to uh, plow the fields, so it's really, really hard, really, really old, and we need to find ways of tenderizing it, making it taste nice. So the first issue is antagonistic pairs. Um, when a muscle moves, it contracts. And then when you move it the other way, it relaxes and it's moved the other way. So things like tenderloin, they're the muscles which are not very active. They are used to move the muscle back. But usually the cheapest cuts of meat are the muscles which are worked very harshly. So that's what makes them very hard and very lean. Stop. If you notice, these muscles are made of fibers, and chewing through the fibers is really hard work. So what we're going to do is when we cut the meat, we're going to cut it this way to cut the fibers. All right. So we'll make sure we cut it like this into steaks like this, and not like that into fillets. Otherwise, it'll still be as hard as it was before. So not like this, but like this. Stop. Start. What we will be doing is we'll be denaturing the proteins using an alkali. Alkalis release OH minus ions, and a protein, it's not just a long thing down there. We want to turn it into long things. It's usually jumbled up. It's got a particular shape. So this will be jumbled up like this, giving it a shape. What holds it together is hydrogen bonds. So if you've got a hydrogen down here in this structure, and you've got another structure there, and what will happen is these two opposite charges will attract, and this holds this together. When we put this, when our alkali, which we're going to be using, calcium carbonate, when it releases these OH ions, this will attract the hydrogen, which is slightly positive, and it will release the other part of the protein, and then it will relax into long thin strands, which are a lot easier to chew. Stop. The problem with alkalis like calcium carbonate is they have a bitter taste, a slightly soapy bitter taste. And we will remove these by something called neutralisation. We're going to add vinegar, which is an acid, which will release the H ions. And when these ions combine, we end up with water. And this water will make our steak slightly more tender. Stop. Most of the steak after we're done. And then what we're going to do is we will add vinegar in excess. So we won't add just enough vinegar to get rid of the OH ions from the calcium carbonate. We'll add a little bit extra. And because the issue is when we fry our steak, the vinegar, the acetic acid in the vinegar, that will mostly evaporate. So we won't end up with a very, very sour taste. Stop. So with the vinegar, we'll add whatever flavorings we need to, whatever marinade we need to. We didn't add it in the beginning because it will cause, if we add too much, it will cause the water from inside the steak to go outside, and the steak will get shriveled and harder. All right. The other thing which we're going to use to make sure the steak is nice and tender is something called osmosis. The cells, there's a cell, it has something called a membrane. The membrane is semi-permeable. meaning some things can cross through the tiny gaps. Things which are small, like H2O. And what will happen is lots of salt in the inside of the cell, very little in the outside, and lots of other things in the inside. So it will go again. It will go with the concentration gradient. So what will happen is water. We will put the steaks inside water, and lots of water will go from the outside of the steak in the inside, making it more tender. And this is because we have not put salt in the water. There's very little salt in the water. We have not added any. We haven't added any flavors yet. And all the we're going to add calcium carbonate, and OH ions will also go through, and the water will go through. So what will happen is that we will notice that the steak will get a lot bigger because it's taking in water. Stop.